Let's start by talking about the DNA. You know that DNA stands for deoxyribonucleic acid. It's a long molecule that is found in cells, mainly in the nucleus. However, some DNA is also found in mitochondria and chloroplasts. Chloroplasts are the organelles, the compartments inside plant cells, which allow photosynthesis and make the plant cell green. So we all know that DNA is actually a polymer. Polymer means it's a long molecule made of attached building blocks, and you can see here DNA is a polymer of what we call nucleotides, and they come in 4 different flavors, and they are defined or called A, G, T, and C, and these are abbreviations for adenine, guanine, cytosine, and thymine. So this colored part, which differs between the 4 different nucleotides, is called the base. And in DNA, the bases of the nucleotides have the property of matching with each other, and they do that very specifically by A binding to T and C binding to G. And we call this the rule of complementarity between nucleotides or between bases, and it allows the formation of a double-stranded helical structure of DNA as you see it in this animation. And this allows 2 strands called complementary strands to have their bases complementing each other and binding to each other, as you might see it here. So the DNA we have in our cells is all the time double-stranded DNA, and this is the short nomenclature used, double-stranded DNA, and it's in this double helix structure. So in this figure it's shown that the polymer of DNA, which is this double-stranded structure, actually allows the coils on itself, and it's compounded by other molecules in the cell called proteins, specifically histones and others, and then it's what makes the chromosome. So a chromosome is a long molecule of DNA that is tightly wrapped around different types of proteins like histones. So the A, T, C, and G encode the genetic information similarly to what you know of like 0 and 1 in computers would encode for the different softwares and programs. The specific sequence on each strand is the code of the genetic information. And as we will come to understand later, if we change the sequence, if mutations happen, well, that could have an impact on how the cell behaves, how the trait appears, and so on.